Hello friends, welcome to Sam Biology class. Friends, today we shall discuss about the remaining two alternative pathways of complement activation. They are the alternative properdin activated pathway and the alternative protease activated pathway. Following this, we shall also discuss as to how the membrane attack complex is assembled from various activated components of the complement system. As you know, the membrane attack complex is a macromolecular complex that directly kills microbes by creating pores in their membrane. First of all, we shall discuss about the alternative properdin activated pathway. While discussing the alternative takeover pathway of complement activation, we came to know about properdin as a regulatory factor in stabilizing the membrane-bound C3 convertase whose composition is C3b-BB. Recent evidences indicate that, in addition to its stabilizing role, properdin could also initiate the alternative pathway of complement activation leading to the amplification. In vitro experiments demonstrate that when properdin molecule is immobilized on an artificial surface and is allowed to interact with purified complement components, then the properdin bind with the component C3b and factor B. By binding, factor B becomes susceptible to cleavage by the factor D. So, the factor B is cleaved into two fragments. Smaller fragment is BA that diffuse away, and the larger fragment is BB that remain associated with the remaining two components to form a new complex that function effectively as the C3 convertase, thus leading to the amplification. So this is the new complex whose composition is C3bPBB. P means properdin. So this new complex also functions effectively as the C3 convertase leading to the amplification. So it seemed that properdin could also initiate the activation of the alternative pathway of the complement system on an artificial surface. Second pathway is the alternative protease activated pathway. Both blood coagulation pathway and the complement system use protease cleavage and conformational change in order to modify the activities of several enzymes, thus leading to amplification of various steps by feed-forward loops. Recent evidences indicate about the existence of functional interaction and theoretical parallel between these two proteolytic cascades. It has been found that blood clotting factors such as thrombin could initiate the alternative pathway of complement activation by inducing the cleavage of complement components such as C3 and C5. So these components C3 and C5 are cleaved to release the active anaphylotoxins such as C3A and C5A. But as this pathway required a higher concentration of thrombin, it was not considered as physiologically significant. But evidences indicate that activation of the blood coagulation cascade could cause the cleavage of a significant amount of complement components such as C3 and C5 into anaphylotoxins such as C3A and C5A respectively. This is about the alternative protease activated pathway. All the three initiation pathways of complement activation leads to the generation of C5 convertase. The composition of C5 convertase is C4b, 2a, 3b for classical and lectin pathway, whereas for the alternative pathway the composition is slightly different. It is C3b, bb, C3b. As you know, C5 convertase cleave the complement component C5 into two fragments. The smaller fragment is C5a and the larger fragment is C5b. The component C5b is generated on the surface of the target cell or on the immune complex and provide a binding site for the subsequent component of the membrane attack complex. The component C5b is extremely labile. Because of this, it is rapidly inactivated unless it is stabilized by binding of another complement component, that is C6. The next step in complement system is the generation of membrane attack complex. All the reactions that we have discussed so far takes place on the hydrophilic surface of the target microbes or on the immune complexes in the fluid phase of blood, lymph or tissues. In contrast to this, when the component C5b binds with the component C6 and C7, the resulting complex, that is C5b67, undergo conformational change that exposes hydrophobic regions on the surface of C7 capable of inserting into the lipid bilayer of the cell membrane. But when the reaction takes place on immune complex or any other non-cellular activating surface, this hydrophobic binding site fails to anchor the complex. Such type of complex is released. Such complexes, that is C5b67, attack nearby cells and mediate their lysis. Under physiological conditions, such lysis is minimized by regulatory proteins. The next component of the complement system is C8. C8 is composed of two polypeptide chains. One is C8b and the other is C8 alpha gamma. Binding of C8b with the C5b67 complex induces conformational change in the C8 dimer. As a result, the hydrophobic domain of C8 alpha gamma insert into the interior of the plasma membrane. This complex of C5b678 can create a pore that can cause the lysis of red blood cells but not that of nucleated cells. The final step in the formation of membrane attack complex is the binding and polymerization of C9 molecules. About 10 to 19 C9 molecules are bound and polymerized by a single C5b678 complex. As a result, C9 molecules undergo transition, because of which they also insert into the plasma membrane so as to form the final membrane attack complex. The membrane attack complex has a tubular form and a functional pore diameter of 80 to 100 angstrom. This membrane attack complex consists of a single complex of C5b678 surrounded by a polyC9 complex. This membrane attack complex directly kill microbes by creating pores in their membrane that leads to their death irreversibly. So friends, this is about complement system. For more updates, please subscribe my YouTube channel Sam Biology Class, and also I'll be very happy if you share your comments and subscribe my YouTube channel. Bye bye, good night.